Artificial intelligence analyzed blood tests can predict the progression of neurodegenerative disease. Blood and brain gene expression trajectories mirror neuropathology and clinical deterioration in neurodegeneration. Most prevalent neurodegenerative disorders take decades to develop, and their early detection is challenged by confounding non-pathological aging processes. For all neurodegenerative conditions, we continue to lack longitudinal gene expression data covering their large temporal evolution, which hinders the understanding of the underlying dynamic molecular mechanisms. Here, scientists overcome this key limitation by introducing a novel gene expression contrastive trajectory inference method that reveals enriched temporal patterns in a diseased population. Evaluated on 1969 subjects in the spectrum of late-onset Alzheimer's and Huntington's disease, this unsupervised machine learning algorithm strongly predicts the neuropathological severity. Furthermore, when applied to in vivo blood samples at baseline, it significantly predicts clinical deterioration and conversion to advanced disease stages, supporting the identification of a minimally invasive blood-based tool for early clinical screening. This technique also allows the discovery of genes and molecular pathways in both peripheral and brain tissues that are highly predictive of disease evolution. 85 to 90% of the most predictive molecular pathways identified in the brain are also top predictors in the blood. These pathways support the importance of studying the peripheral brain axis, providing further evidence for a key role of vascular structure functioning and immune system response. The gene expression contrastive trajectory inference is a promising tool for revealing complex neuropathological mechanisms with direct implications for implementing personalized dynamic treatments in neurology. Scientists at the Montreal Neurological Institute and Hospital of McGill University and the Ludmer Center for Neuroinformatics and Mental Health used an artificial intelligence algorithm to analyze the blood and post-mortem brain samples of 1969 patients with Alzheimer's and Huntington's disease. Their goal was to find molecular patterns specific to these diseases. The algorithm was able to detect how these patients' genes express themselves in unique ways over decades. This offers the first long-term view of molecular changes underlying neurodegeneration, an important accomplishment because neurodegenerative diseases develop over years. Previous studies of neurodegeneration often used static or snapshot data and therefore are limited in how much they can reveal about the typically slow progression of disease. This study aims to uncover chronological information contained in long-scale data by covering decades of disease progression, revealing how changes in gene expression over that time are related to changes in the patient's condition. Furthermore, the blood test detected 85 to 90% of the top predictive molecular pathways that the test of post-mortem brain data did, showing a striking similarity between molecular alterations in both the brain and peripheral body. This test could one day be used by doctors to evaluate patients and prescribe therapies tailored to their needs. It could also be used in clinical trials to categorize patients and better determine how experimental drugs impacted their predicted disease progression. The next steps will be testing these models in other diseases such as Parkinson's disease and amyotrophic lateral sclerosis. The study was made possible with data made openly available through the Alzheimer's Disease Neuroimaging Initiative (ADNI) and the Rush Alzheimer's Disease Center, Rush University Medical Center, Chicago. Thank you for watching the video. If you like the video, don't forget to like and follow my page so that you can get other updates in the future. Thank you.